way have been published, Dalbergia latifolia and Dalbergia siso, but the protocol obtained were still not promising for plant propagation. The proliferation and shoot grow pattern generally did not appear as prolific as those of other hardwood species such as teak. For culture derived from mature plant, the proliferation and growth of shoot were limited, and the growth of rooted shoot looked weak. Research on rootwood suspension culture has not been done much, while callus induction for organogenesis from previous studies is still not optimal. So in this research, I will study about suspension culture. Material and method. Plant material, we use friable callus of accession Ng6A from semi-solid media. Then the initiation of rosewood cell suspension culture, the 5 mg of 2,4-D and 3 mg picoram, and 10 mg 2,4-D and 10 mg NAA. And then the proliferation of cell suspension culture in 5 mg 2,4-D and 3 mg picoram. Then cell anatomy observation, and then the effect of 2,4-D and picoram on the proliferation of round and long cell. The combination treatment of 2,4-D 0, 5, and 10 mg and picoram 0.3, 1, and 3 mg. The result, the initiation of rosewood cell suspension culture, we can see from figure 1, the growth of rosewood cell suspension is high in 2,4-D and picoram, then 2,4-D and EA. And according from table 1, we can see that 2,4-D and picoram have the high number of cell, but the round cell is low. This is different with 2,4-D and NEA, is the number of cell is low and the round cell is high. But we focus on proliferate, so the next experiment is 2,4-D and picoram. This is the figure of 2,4-D and EA, and this one is the 2,4-D and picoram. You can see that the callus proliferation. This is the morphology cell of shells in suspension callus. You can see it's 1A. This is the round cell with concentrated cytoplasm, and this is 1B, round cell with non-concentrated cytoplasmic, and this is the elongated cell. This is the cluster with round and the compact structure, round cell. This is the cluster with large vacuola. The size effect of cell cluster inoculum on the proliferation of rosewood cell suspension. Table 2, we can see from table 2 the inoculum size 1 until 2 mm. The browning is high and the proliferation is low. And but the less than 1 mm is no browning and the proliferation is high. We can see in figure 3A, we can see this is the browning cell and not proliferate with inoculum size 1 until 2 mm, and B less than 1 mm, we can see no browning and the cell is proliferate. The cell anatomy, this is the cell of callus of rosewood, and this is the histology of cell. We can see in number 1, the auto red is, this is, the cell is big parenchyma, and the number 2 and number 3 is begotten to proembryonic mass, and we can see the curve line. This is one. We can see is this is the line is resemble boundaries, the structure round structure in proembryonic mass. This is the cell with the morphology cell with azotocarmine. We can see in arrow red, this is the proembryonic mass, and in B, C, and D is the subculture 5, 6, and 7. We can see this the cell cluster from the subculture. Okay, the last effect of 2,4-D and picoram on suspension cell proliferation and cell morphology. Table 3, we can see that the cell suspension proliferate decrease with increasing concentration of 2,4-D from 0 to 5. And in the medium without 2,4-D, the highest proliferation was achieved in 0.3 milligram picoram and decrease with increasing picoram concentration. The conclusion, cell suspension culture of rosewood could be induced and established from seed-derived callus maintained on a semi-solid medium. An inoculum of 0.1 gram consisted of cell clumps smaller than 1 millimeter continue to proliferate and form suspension. Even though slowly in MS liquid medium supplement with 5 milligram 2,4-D and 3 milligram picoram. The rosewood cell suspension evidently consisted of elongated parenchyma cell, isodiametric cell, and cluster of cells that resembled proembryonic mass, which one the proliferate unit of embryonic culture. Attempt to optimize the growth of development of cell suspension disclose that inoculum of cell cluster less than 1 millimeter resulted in continued growth without any browning, while picoram supported the division and proliferation of the suspension with high percentage of elongated cell, and 2,4-D caused differentiation of isodiametric cell important for the development of embryonic culture. Acknowledgement. This research was funded by the Badan Riset BRIN program and DIPA, Research Organization for Life Sciences and Environment. And thank you for your attention.
Okay, thank you for interesting presentation. So we are going to go to the next presenters. It will be presented by Mr. Risang Pandegan. The title is Isolation of Aquaporin, Encoding Gene from Cassava Manihot Esculenta Crans, Genotype Ubi Kuning, Induced by Drought Stress. So let's enjoy.
Today we will present our research with the title Isolation of Aquaporin Encoding Gene from Cassava Manihot Esculenta Crans, Genotype Ubi Kuning, used by Drought. Introduction. Cassava is an important crop in Africa, Asia, Latin America, and Caribbean. It can be grown with minimal inputs, but gives considerably higher yields with fertilizer and good management. The ability of cassava to tolerate drought and soil with low fertility farming in dry areas in various parts of Indonesia daily substrate and for small scale farming. Although cassava is one of the most tolerant crops, the mechanism underlying its ability to survive and produce under drought remains brain strength resistance mechanism consists of the first, the genes that play a role in cell protection with stress, controlling gene in pressure, repair and detoxification, and adaptation. Second, the genes that regulate the expression of other genes involved in stress response, such as signal transducer genes, kinase and phosphatase, membrane intrinsic protein, MIP, like aquaporin and UF, transcription factor. Since in tissue water potential are related to aquaporin that play a role in controlling the efficiency of water transport to cell membrane during a water deficit. Two, figure one, aquaporin, plus model, first signature, and ultimately aquaporin, in the form of activating pollen element. The motif would be during phosphorylation type also present at the carboxyl and hydrogen and liquid. And in liquid, there is a histidine residue which involved in solid residue. The aim of the study was to isolate the aquaporin gene and therefore first transcription PCR technique with specific primer can drug with copy DNA. Second, to identify the homology of transitive fragment of drug-tolerant cassava aquaporin gene based on uniform difference. Third slide, material and plant material. Cassava genotype ubiquinum. The plant was grown in plastic pot, 20 cm to 25 cm, containing mixed field, soil, and sand, with temperature 1:1 and water once a day with 550 milliliters of water in the morning. 3-month-old plants was used to water stress treatment. Water stress treatment was used in the greenhouse by decreasing the water volume from 550 milliliter control to mild stress 37 milliliter and severe stress no water. Stress treatment period during 26 days. Figure 2 shows cassava genotype ubiquinum with water control on A side and dose treatment on B side. We did total RNA extraction of cassava leaves and then copy DNA synthesis, PCR, and the last for sequence analysis and homology of NEAP. The third leaf from the top of the control plant and the retained leaf of the stress plant was used for RNA isolation plant material. 5 pairs of primer were used to amplify the cDNA of the ubiquinum control and water stress plant. 2 of the primers were degenerate primers. We did 35 cycles with annealing, elimination, and post-PCR in PCR conditions. Table 1 shows primer and sequence to amplify the DNA of cassava designed with an NDACP AU 5992241. We name the first primer pair as aquafor 1, primer pair 2, aquafor 2. There were degenerate primer. And third primer pair as aquafor 3, fourth primer pair as aquafor 4, and also aquafor 5 for primer pair 6. For PCR station, the result samples UK1 control plant and U1 water stress treatment can be amplified with aquafor 3 and aquafor 4 at different temperatures. Figure 3 shows the artificial amplification. UK1 use aquafor for 3 and aqua for 4. U1 with aqua for 3 and aqua for 3 with annealing temperature at 56.9 degrees Celsius and 57.6 degrees Celsius. For sequence analysis, the alignment results between concentric complete sequences showed high homology between the NEAQB fragments of Ubi-Kuning treatment, control, and NEAQB AU. Discovery query 1999. The differences in nucleotide phase shown red buffer for the 100 events between 70 and 190 until 800. There were two similarity percentage of NEAQB sequence. Similarity percentage between samples, control, and treatment, 98.28%, 199.83%. And then between samples, control, and treatment, and reference, NEAQB AU was 98.16%, 99.51%. For cluster analysis, control plants, NEAQB 1.1, and group stress treated plants, NEAQB 2.1, are in the same cluster as the reference AQB sequences, which was NEAQB AU 59222.1. The phylogenetic tree of 4 NEAQB samples of nucleotide based on neighbor joining reconstruction method was shown in figure 5. Ubi-Kuning reduced amino acid sequences have evolutionary rates that are close to PIP, the plasma membrane protein. Figure 6 shown the conclusion of this research. First, NEAQB1 was successfully isolated from cassava Ubi-Kuning, treated with drought stress for 26 days, and showed highly homologic to the aquaporin gene of NEAQB AU. Second, putative aquaporin fragment can potentially reduce in the genetic improvement of drought-tolerant cassava. Many thanks.
Okay, thank you for the interesting presentation about the aquaporin from Manihat Esculenta. So we are going to the next presenters. The next presenter is Ms. Fikrinda about improvement of insecticides biology properties under soil phosphorus manipulation by indigenous cellulosic fungi and rich organic fertilizers. So let's start. Thank you.
Thank you, moderator. Good morning, good afternoon, everybody. I would like to present our paper. The title is Improvement of Insecticides Biological Properties under Soil Phosphorus Manipulation by Indigenous Cellulosic Fungi and Rich Organic Fertilizers. Our team, I'm Fikrinda, Mr. Munawar Khalil, and Mrs. Erita Hayati. We are from Universitas Syakuala, Banda Aceh, Indonesia. As a background of this study, we know soil biology is an essential component of soil quality. They may contribute to many soil functions such as carbon regulation, nutrient cycling, water retention, aggregation, pH buffering capacity, and disease and pest management or regulation. The communities in the soil depend on nutrients. Phosphorus is an essential nutrient affecting soil microbial population and activities. The organic fertilizers provide nutrients to soil organisms, but the quality of the organic fertilizer influence its effectiveness as a soil amendment. Amending decomposer microbes such as cellulosic fungi may improve nutrient content of the organic fertilizer. The objectives of the study were first, we want to assess the response of soil microbes, respiration, and available phosphorus to cellulosic fungi enrich organic fertilizer addition. And then we also want to identify which soil biological properties are correlated with available phosphorus. The experimental design we use 7 treatments without organic fertilizer or control, 5 and 10 gram per kilogram compost, 10 and 5 gram per kilogram co-manure, and 5 and 10 gram per kilogram vermicompost. The treatments were replicate 3 times, and we set the experiment for 2 series. The procedures of this experiment: first, we prepare soil medium preparation, and then we prepare cellulosic fungi inoculant. The isolate we use was Penicillium sp isolate SR 18 from University Farm of Banda Aceh. And then we prepare an incubation the cellulosic fungi enriched organic fertilizer for 30 and 60 days. After the incubations, we analyze some soil characteristics such as available phosphorus, total microbes, phosphate solubilizer microbe, and respiration. The data we have, we analyze statistically by SPSS version 25, and the difference among the treatments we analyze by Duncan method. The result and the discussion here we can see the soil characteristic before we use we examine the treatment. You can see texture was clay loam, pH neutral, organic carbon and exchangeable potassium were low, total nitrogen and cation exchangeable capacity were medium, total phosphorus was high, and available phosphorus was very high. For the cellulosic fungi organic fertilizer we use in this study, we can see that vermicompost has highest in nitrogen, phosphorus, and potassium contents. The result in this experiment for available phosphorus, we can see that the highest available phosphorus we found at the 10 gram per kilogram vermicompost at 30 days after incubation. But at 60 days after incubation, the best treatment was at 10 gram per kilogram co-manure. For total microbes, we can see that 3 treatments at 30 days after incubation, i.e. 5 gram per kilogram co-manure and 5 and 10 gram per kilogram vermicompost while at 30 days after incubation, the best treatment were at 2 treatment, i.e. 10 gram per kilogram co-manure and 10 gram per kilogram vermicompost. For phosphate solubilizer microbes, the best effect was at 5 gram per kilogram co-manure, but the treatments that different from other treatments, i.e. 10 gram per kilogram compost, 10 gram per kilogram co-manure, and 5 and 10 gram per kilogram vermicompost at 30 days after incubation. But at 60 days after incubation, three treatments have had similar effect to give best result for phosphate solubilizer microbe, i.e. 5 gram per kilogram manure and 5 and 10 gram per kilogram vermicompost. And the last, for respiration, the best effect we found at 10 gram per kilogram compost, co-manure and vermicompost at the same number, the same amount for at 30 days after incubation, and at 60 days after incubation, the best treatment was 10 gram per kilogram vermicompost. The relationship between soil available phosphorus and soil biological properties, we can see that we found that soil available phosphorus has a positive relationship to total microbes at 30 and 60 days after incubation, and phosphate solubilizer at 30 days and 60 days after incubation, but for respiration just at 30 days after incubation, but not at 60 days after incubation. The conclusion, we conclude first, the use of indigenous cellulosic fungi enriched organic fertilizer increase soil available phosphorus and microbial population and activities. And then soil available phosphorus has a positive correlation with the soil's biological properties. So, and that's all. Thank you for your attention.
Thank you for the interesting presentations about fertilizers, improvement of the insecticides, biological properties under soil phosphorus. Okay, so wait a minute. So the next presenters, it will be our last presenters. So welcome to Miss Lina Herliana. She will present about screening Plantago ovata, psyllium, gamma irradiated putative mutants for higher mucilage yields. So the time is yours. Thank you.
Good afternoon, everyone. First of all, I'd like to thank the organizer of ISIBio 9 2022 for selecting my oral presentation. Let me introduce myself. I'm Lina Herliana, a scientist at OR Hayati dan Lingkungan, Pusat Riset Rekayasa Genetika BRIN. Today, I'm presenting one of my works during my PhD at the Burton Lab, University of Adelaide in Australia. My presentation title today is Screening Putative Plantago ovata Psyllium Gamma Irradiated Mutants for Higher Mucilage Yields. My talk is particularly relevant to those looking for a simple but powerful screening method. Let's begin with the introduction. Plantago ovata is an annual plant that produces seed husk called psyllium. It turns into gel-like structure called mucilage when wet. It has a many downstream application. It has been known as a medicinal plant, as a dietary fiber to reduce constipation and lower blood cholesterol levels. You may be familiar with supplement brands Metamucil or Flimty that have psyllium as the main ingredient. Psyllium is also widely used as a gluten replacement in bread making. As the industry grows, the demand for psyllium increases. India dominates about 80% of the global market for psyllium. The productivity of an Indian variety is negatively affected by adverse environmental conditions, and it causes unstable prices. Limited success has been reported in improving productivity due to a narrow genetic base. Variation can be achieved by exploring wild relatives or by induced mutation. My supervisor, Professor Rachel Burton, with her team in Australia, is establishing a breeding program for this plant. They generated gamma-irradiated mutant. My study focused on selecting a Plantago mutant line with a higher mucilage yield than the wild type, but with no developmental defects. Let's move to the material and the methods. The 201 putative mutant lines had been propagated by selfing to the 5th generation. I randomized and split sample into 12 batches as the processing took several days. Before mucilage extraction, 20 seeds for each mutant line were weighed using an analytical balance and photographed under a dissecting microscope to obtain seed measurements. To identify the mucilage extrusion pattern, representative seeds from each line were stained with ruthenium red RR. I extracted mucilage from seed using a thermomixer and then poured the mucilage into 96 well before freeze drying the samples. Then dry mucilage was weighed to obtain mucilage yield. I use a Fiji image J to process images and MS Microsoft Excel and R to run the analysis, including correlation analysis, principal component analysis, student t-test, and one-way analysis of variance ANOVA. Okay, the next 5 slides will be result and discussion. Variation of mucilage extrusion pattern can be found in a gamma-irradiated mutant lines. Tucker et al. 2017 used the same source of mutant where the seed phenotype were linked to variation in gene expression of heteroxylon biosynthesis, which is the main component of Plantago mucilage. Our study focused on mucilage quantity rather than mucilage quality. However, there is no link between the amount and the extrusion patterns. Therefore, we investigate the seed weight and other seed dimensions in the following slides. Figure A shows that a large proportion of gamma-irradiated mutant line had a reduction in the seed weight. This trend was also observed as a reduction in a mucilage yield per seed in a figure B. About 68% of the mutant had reduced mucilage amounts. Figure C, the proportion of mucilage yield per dry seed weight shows the opposite trend. More mutant lines have a higher proportion of mucilage, about 73% of the population. We then ran a correlation analysis to confirm the relationship between mucilage yield and the seed dimension. Figure A shows the correlation matrix with a blue meaning positive, while orange means the negative correlation between two variables. Color intensity and the size of the circle are proportional to the correlation coefficient. 0 means no correlation, while 1 means strong correlation. Mucilage yield per seed in a figure or table B had a positive and robust correlation with the seed weight, area, perimeter, and diameter. It also had a positive but weak correlation with the mucilage yield per area and circularity. No significant correlation was seen between a mucilage yield per seed and roundness and mucilage yield per dry seed weight. In contrast, mucilage yield per seed weight was mostly negatively correlated with the seed dimension variables. While a correlation plot helps investigate pairwise correlation, principal component analysis reduced data dimension. 9 variables were reduced into 2 principal components, PC1 and PC2. These 2 PC explain 97% of the variation in the data. Individual sample, wild type, and mutant line were visualized in plot with 3 different color groups. Figure A was colored according to seed weight, figure B by mucilage yield per seed, and figure C by mucilage yield per dry seed weight. Samples with similar profile were grouped. Each variable, seed weight, mucilage yield per seed, and dry weight, had 4 groups, from the lowest in group 1 to the highest in group 4, by comparing the mutant lines to the wild type value for each measurement. For this study, the candidate mutant needed to have similar seed phenotype characteristic to the wild type, with no grow habit defect. The only affected differences being the mucilage yield. We then measured seed and mucilage yield per plant with 8 replicates from line in categories 3 for seed weight, 4 for mucilage yield per seed, and 3 for mucilage yield per dry weight. We selected 970-1, as you can see in the figure with the red arrows, as the best line producing more mucilage with the lowest variation among the plants. It was named Raya, Ray, which means a greater in Indonesian. Wild type and Ray plants grew healthily and no distinct phenotype were observed in figure A. Figures B and until D shows no significant differences between plant dry weight, seed weight, and harvest index between these 2 genotypes. In figure E and F, overall, the mucilage yield of Ray grown either in the grow chamber or in the field was higher than the wild type grown at the same time and under the same condition. However, mucilage yield per dry weight was significantly higher from a plant grown in the field, while mucilage yield per seed was considerably higher from plant grown in the grow chamber. Okay, I'm now nearing the end of my presentation. Let me just run the key point of this presentation. Gamma irradiated mutant provide valuable resources for genetic improvement. Seed dimension, especially seed weight, affect mucilage yield. Many combinations of seed characteristics can be used to select mutant according to the end use. In this study, mutant line was screened to obtain 1 candidate mutant suitable for identifying gene related to mucilage production. The mutant was named Araya. Before Q&A, I'd like to acknowledge Dr. Tucker, Dr. Pan, Sivanghor, and Tiko Newman for their hard work generating Plantagovata mutant population, and Dr. Tina Bianco Mioto for continued support. And I was supported by the University of Adelaide, Adelaide Graduate Research Scholarship and BPPT, and now the National Research and Innovation Agency, BRIN Indonesia. Thank you very much.
Okay. This is the end of the presentation. So thank you for all very interesting.
Termute, Mbak.
Yeah, sorry. Yeah, okay. So we are coming to the discussion session. Now you can ask questions by raise your hand or using the chat feature. Is there any questions? Or I will start with the questions for Yeah, I will start the question if there is no questions from the audience. Okay, so first my question is for the first presenter, for Ms. Wasiatu Roziko. Are you here? Okay, so thank you for your interesting presentations. I have a questions. So you are doing cytological characterization of the chili that's already had the gamma irradiation. So my question is, according to your conclusion and the data that you have so far about the cytological and the characteristic, and then what is your overview for the next for the future of your research? Like your future plan for this research.
Can you repeat the question?
Okay, so I will make it clear. So I remember from your data, it shows that in general there are some defects in stomata after the irradiation, right?
Yes, yes.
Yeah. So regard to this, for example, what is your overview or your plan for the future research regards to this data?
Also from the cytological characteristic, we see the result is randomly. So the next step is myself will doing the through the
Probably you can because the stomata probably relate to the photosynthesis or other aspects that we already know so far. So probably for the next experiments, you can plan about photosynthesis or another things. That's what I mean. Do you have any plan about that?
So my plan is research in growth stress.
Pardon?
Growth stress.
What is this?
Taman kekeringan.
Okay, I'm sorry, but I don't really catch the voice.
So my plans for this research, I will continue with growth stress.
Oh, okay, yeah. Okay, thank you. Because this is the preliminary data, so probably you can develop further for your further research. Okay. Is there any questions from others? Okay, I have questions then again for the next presenters. This is really interesting for me because this is not really my field, yeah. But Bombix Mori or the silks productions, I think this is also important. And so I want to ask to the Ms. Retno Agustarini. So based on your presentations, the data was obtained on 2017. So can you give us an overview about the current situation about this project today?
Okay, thank you. Thank you. Yes, Bombix Mori is producer of silk. Silk is, as we know, silk is a natural fiber in the form of filament of cocoon of the silkworm Bombyx mori. For your question about how this research now, as I asked in the challenge in the development of sericulture in PFMU Balimo, there are marketing and policy barrier in its development. The ability of the lack of motivation and commitment of the leader of PMMU to continue the program is very sorry. The commitment of the policy implementer are some of the key of the successful implementing in the program. But the lack of motivation and commitment of leader of the PFMU Balimo is no longer running sericulture development in PFMU Balimo. So in this year, sericulture is no more running well in the PFMU Balimo.
Okay. So that's really unfortunate, yes.
Yes.
Actually, is it possible that, for example, we do not sell the product as cocoon, because I think that the cocoon is the raw materials. Can we, for example, process it until at least minimum to the medium materials, not the final product, but the medium? Probably it will help the economic, it will increase the economic value for this product.
Yes, in this paper, we in the beginning of the plan, the PFMU Balimo is Balimo with the sorry. PFMU Balimo with a mediator of FRDC researchers began to initiate marketing with Kurnia Sham. This is a fifth generation owner of Arni Kurnia, producer of Sengkang seed weaving South Sulawesi. And next, the PFMU Balimo is also produce the cocoon of the FSG production into the yarn, and it can improve the price of the cocoon.
Okay. Okay, thank you very much for your answer and the nice discussion. So hopefully in the future there will be more progressions in the sericulture in Indonesia.
Yes, thank you.
Okay, I saw that there is somebody who raised a hand.
There is a question in chat.
Oh, there is a question in chat. Yeah, okay, I'm sorry. Okay, so there is a question for first is for Ms. Wasyato Raziko. How much LC50 dose of radiation treatment of chili plants? Does each cultivar have a different LC50 doses? Is there any specific treatment for selecting mutants that have good agronomic characters? So you can answer first.
Okay, thank you. I will answer the question. How much does LC50 dose of radiation treatment of chili plant? So in my research, we are not measure the LC50 dose, but we are only according to agronomic characteristics. So we are not measure the LC50 dose. And then the next question is, does each cultivar have a different LC50 dose?
So the cultivar which have a different LC dose is Kokai, Laris, and SLC. And the next question, is there any specific treatment for selecting mutant that have good agronomic characters? So in my research is nothing which are the treatment for selecting mutant. So only agronomic observation and cytological observation.
So continue with the next questions, because this is also to Ms. Wasiaturozikoh. So have you tested the mutant chili in abiotic stress conditions?
Okay, I didn't have a test the mutant chili, so only observation in agronomic and cytological characteristics.
Okay, thank you. So there is a question for Ms. Retno Agustarini. How about the silkworms, eggs, and process product packaging and maintenance to stay at long duration of expired date? So please.
Can you repeat?
So this is the question from Mr. Risang Pandegan. He's asking about how about the silkworm eggs and process product packaging and maintenance to stay at long duration of expired date. About the egg of the silkworms, how to maintain to maintain it so that it can stay longer. Can you answer it, Ms. Retno Agustarini?
Okay. Maintain the egg of silkworm.
Yeah.
We usually keep. We usually keep the egg and store it in the refrigerator with specific
Temperature
in 5 and 2.5 degree. This is called save the egg silkworm until about 80 to 60 to 80 days.
Okay.
The temperature, and we usually sorry, for the silkworm egg and process product packaging.
Okay.
We should usually send this egg with the paper of we call it a paper to the
what man paper?
Egg paper. To the customer. Yes. Okay.
Okay, thank you very much. So actually there is another 2 questions, but the time is up. So I will just read the questions. So probably if you want to know the answer further, you can contact the presenters. So So the present actually Mr. Risampadegan give some questions. The questions about what are the difficulties when do self-suspension culture for hardwood species. Yeah, this was the presenter for Miss this is direct to Miss Indira Riastiwi. What are the difficulties when do self-suspension culture for hardwood species? And there is also another questions for presenter number for Miss Lina. What kind of thermomixer condition that you use to extract mucilage, and naturally what's the function of mucilage for the seeds? Okay, so Miss Helena already answered it.
No, actually, it's just the method. We already published it, only the method. So
okay.
I put it in there
Yeah.
for the link for Parisang to maybe can look further. Yeah.
Yeah. Okay. So actually, also there is a question from Miss Sri Hartati. Questions about Tubulina again. In general, what type of the major component of Plantago ovata mucilage is it a kind of polysaccharides?
Yeah.
And
do you want? Oh, sorry. Do you want
Yeah.
me to answer this question quickly?
Okay, if it is short.
Okay.
Yeah.
Yeah. So mainly, Plantago ovata consists of polysaccharide, is heteroxylon or xylon, and there is a mannose. I mean the monosaccharide, this rhamnose and mannose, and it's and pectin. But in Arabidopsis, mostly pectin. In another like linum is balance between pectin and heteroxylon or xylon. But in Plantago, the majority is heteroxylon or xylon. Yeah, it's different plant. They have different, but I mean the general polysaccharide, xylon and pectin. But the composition will be different in plant, including in Plantago genus itself.
Okay.
Yeah.
Okay. Thank you very much. I'm sorry if. Thank you very much for all the presenters, all the for the discussions. I'm sorry if some of the presenters cannot answer the questions because of the time. So I'm sorry if I have some mistakes or anything. So I will end this session. Thank you very much. So go back to the committee. Mbak Fitri udah selesai ya? Oke, tak pikir nanti tuh harusnya ada, saya pikir ada ini closing dari panitianya, nggak ada ya? Iya, nggak ada ya, Mbak? Saya tadi belum aku tutup, ya udah nggak apa-apa. Ya, Assalamualaikum warahmatullahi wabarakatuh.
Welcome back to the main room, ladies and gentlemen. The next agenda is plenary sessions for this afternoon. Therefore, I may invite Dr. Ira Handayani as the moderator of these sessions. But before that, allow me to read short profile of Dr. Ira. Dr. Ira Handayani is currently a researcher in Research Center for Applied Microbiology, National Research and Innovation Agency, the Republic of Indonesia, who are interested in molecular genetics, microbiology, and bioinformatics. She obtained her bachelor degree in chemistry at University of 11 Maret, and she continued her master degree in biochemistry at Bandung Institute of Technology, or ITB, and also her PhD in chemical engineering at Tübingen University, Germany. Dr. Ira, time is yours to lead the sessions.
Okay, thank you, Ms. Aulia. Are you hear my voice? Okay. Hello, are you hear my voice?
Yes, we can hear you
Okay.
clearly.
Okay.
Dr. Ira.
I don't know whether Yvonne already in here or not.
Yes, good afternoon, Ian.
Oh,
I
good
hear you,
afternoon,
and I'm
Yvonne.
already here.
Okay, how are you?
Thanks, fine. Hope you too.
Okay. Okay, thank you. Okay, I will start the presentation. The plenary session for this seminar. First of all, I'm very happy to see you here and welcome all of you to this plenary session. Before the presentation starts, let me tell you how the presentation will go. First, our first speaker, Professor Yvonne Mas, will be invited to present the material, and then our second speaker, Professor Claudia Tiribelli, will also be asked to present his material. After both speakers present their material, there will be a question and answer session followed by a conclusion. Now let's start with our first speaker, Professor Dr. Yvonne Mas. Okay, this is the CV of Professor Dr. Mas. Professor Dr. Mas is a professor of applied microbiology at the Technical University of Brunswick and head of the Department of Bioresources for Bioeconomy and Health Research at the Leibniz Institute, the SMZ, a German collection of macroorganisms and cell culture. She has experience as an editor in the journal Molecule and the journal Printer in Microbiology, and experiment as a member of the topical advisory panel in the Journal of Antibiotics. She is interested in research in the field of microbiology, biotechnology, actinomycetes, antibiotic, and regulation. Now allow me to welcome Professor Dr. Yvonne Mas to make her presentation. Professor Mas, please explain your material. And time is your Yvonne.
So thanks, Ira, for this nice introduction, and good afternoon to everybody.
Okay.
Okay, now I see my slides. Okay. Okay. So again, good afternoon to everybody. Yeah, thanks for the nice introduction. Actually, I'm here at the Leibniz Institute, DSMC, so the German collection of microorganisms and cell cultures in Braunschweig, Germany. And it's a great honor to give a talk during your session at the International Symposium of Innovative Bioproduction in Indonesia. And today I would like to introduce you into the topic of how we can exploit the biodiversity for novel natural compound production by using regulatory switches. So I should, now it's okay. So before I go into that more in detail, I first want to mention why this kind of research is relevant at all. And I think all of you know that antibiotic research indeed is relevant because we observe growing crisis from antibiotic-resistant bacteria. So the World Health Organization regards the antibiotic resistance disease as one of the 3 biggest threats to global health. So of course, it's urgently needed to find novel antibiotics to combat these drug resistances. But the question is where to find them. I'm sorry, the presentation is a bit slow, but I hope we can do that.
I think it's okay.
Okay. So we can, of course, find antibiotics a lot in actinomycetes. So actinomycetes are known to produce 2/3 of all antibiotic in clinical use today. And this antibiotic production potential is known for quite a long time. However, what is not known for such a long time is that the genetic potential for the production of antibiotics is even larger in these organisms. And this appeared when the first genome sequence of such an antibiotic producer has been sequenced. So this was the genome sequence of the model organism, Streptomyces silicolor. And that silicolor strain is known to produce up to 2 to 4 secondary metabolites under standard lab conditions. However, the genome sequence analysis revealed that the genome harbors up to 20 different biosynthetic gene clusters. However, not all of those gene clusters are expressed, meaning not all of these substances are really produced, but a lot of them occur as so-called silent antibiotic gene clusters. And this is not only true for that organism, but nowadays, since we have a lot of, more than hundreds of genome sequences available from different actinomycetes, it turned out that this is true for a lot of these producing organisms. So obviously, actinomycetes still hold a huge potential for the production of novel secondary metabolites. And of course, the current trend is to activate these kind of silent gene clusters. And now I would like to introduce you into the topic, how we can activate those kind of silent gene clusters in actinomycetes. Okay, and we actually can do that by using regulators. And regulators indeed are the set screw to unlock the biosynthetic potential of these natural compound producers. However, the problem is just that there are so many different transcriptional regulators abundant in actinomycetes, especially in streptomycetes, and these can be of different classes, and they can interact in quite complex cascades with each other. And depending on genome sizes, those can actually be more than thousands of different transcriptional regulators. So the question is, which is the most suitable and most potent type of regulator that is suitable to be used as a general activator of antibiotic biosynthesis. And today I will focus on these subtype regulators, since we think that these are good candidates to be used as general activators of antibiotic biosynthesis. Okay, presentation is still slow, but okay. So first of all, I will introduce you where we found this type of regulators. And we indeed identified them in the antibiotic producer Streptomyces spiralis, which is kind of our working horse. So this strain produces the pristinamycin substance, the streptogramin antibiotic pristinamycin. And pristinamycin is actually 2 different types of antibiotics, the peptide antibiotic pristinamycin 1 and the polyketide antibiotic pristinamycin 2. And the Streptomyces spiralis strain produces those 2 substances in a specific ratio. They interact synergistically with each other, and a semi-synthetic derivative of that substance is on the market. It's called Synergid, and it's used as an antibiotic of last resort against highly resistant pathogens. And what we did back then was that we characterized the antibiotic biosynthetic gene cluster. And back then, this was the largest antibiotic gene cluster that has been described so far. And the genes that you can see here in red, they are needed to, they encode for the peptide antibiotic pristinamycin 1, and the blue ones encode for the polyketide antibiotic pristinamycin 2. So when we had a closer look into this antibiotic gene cluster, then we identified a set of 7 different transcriptional regulatory genes, and they belong to different types of regulatory families. And actually, this is quite a large number, because usually these kind of antibiotic gene clusters have one or two such kind of regulatory genes. And we studied the function of all of those regulators in detail. However, today I will only focus on this red labeled PAPR2 regulator here, which belongs to the subtype family of regulators. So when we studied the function of this regulator more in detail, and there I need the next slide. Okay. First of all, we inactivated the gene and studied the pristinamycin production, and there we observed that the inactivation of the PAPR2 gene leads to complete loss of pristinamycin production in comparison to the wild type, which still nicely produced pristinamycin 1 and pristinamycin 2. So therefore, we knew that PAPR2 is an essential activator of pristinamycin biosynthesis. Ira, can I ask, are you the one who is moving the slides forward, or can I do that?
No, I don't know.
Okay,
No,
because it's.
I think it's a bit left in there.
Yeah, it's really several.
I will ask Sandy.
But I will. It's okay. I will just move on. So PAPR2 is an essential activator of pristinamycin biosynthesis. This was what I wanted to say. And we studied the function of that regulator also more in detail, and figured out where the regulator really binds within the antibiotic gene cluster. So therefore, we purified the PAPR2 regulator and did electrophoretic mobility shift assays, and thereby we could show that PAPR2 really binds to the promoter regions in front of the pristinamycin 1 and pristinamycin 2 structural genes. And we were also able to define a specific consensus sequence for such a binding. And by RT-PCR experiments, which are shown here at the right-hand side, we also could show that there's no transcription of the pristinamycin 1 and pristinamycin 2 structural genes in the PAPR2 mutant in comparison to the wild-type organism. So therefore, we knew that PAPR2 is a direct activator and an essential activator of pristinamycin biosynthesis in Streptomyces pristinus pruritus. And the next slide, which I hope comes in a few seconds, shows you where the regulator is situated within the regulatory signaling cascade. Because it's here located at the very bottom, it directly activates the pristinamycin biosynthesis, and itself it is regulated by a set of other transcriptional regulators where I will not go in detail. However, it's quite complex signaling cascade governed by microbial hormones called gamma-butyronolactons. And what I wanted to mention here is that understanding these kind of regulatory signaling cascades and the regulation mechanisms of antibiotic biosynthesis in general can also help to activate silent gene clusters. And this we observed when we overexpressed the PAPR2 regulator in a foreign strain. And back then, this was Streptomyces libidans. And this libidans strain should come also in a second. This libidans strain usually does not produce any secondary metabolite under standard lab conditions. So this you can see here on the left-hand side, the libidans strain with the anti-vector control. However, when we overexpress the PAPR2 regulator, then the libidans strain produces an intensively red pigmented substance. And this red pigment indeed is andecyl protegiasin. And andecyl protegiasin is a known product from Streptomyces libidans. However, it is usually not produced in the lab, so it's encoded by a kind of a silent gene cluster. And this andecyl protegiasin biosynthesis is known to be under control of a subtype regulator called RED-D. And this RED-D shows quite high sequence homology to our PAPR2 regulator, so up to 55% sequence homology. And therefore, we speculated that PAPR2 takes over the function of RED-D and leads to the activation of this silent andecyl protegiasin biosynthesis in Streptomyces libidans. And we also later on could show that on the molecular level. So I'm really sorry that it's hanging, but yeah, this is what is shown in here. So having that in mind, we had a closer look into this kind of subtype regulator families in general. So in literature, they are described as streptomyces antibiotic regulatory proteins. They are known to act as pathway-specific activators of antibiotic biosynthesis. And all of them contain a conserved N-terminal DNA binding domain. And with this, they bind to conserved consensus sequences at the promoter DNAs of the promoter regions in front of the target DNAs. However, so far, the regulatory mechanisms, how those regulators really work, is totally unclear. So this is something what we are going to study in the near future. So we will do crystallizations of the regulator and try to figure out how this activation mechanism really works out in the antibiotic producers. So having that in mind, we had a closer look where those subtype regulators occur, and we found that the genes are very abundant in many different actinomycetes. So you can find them in nearly all streptomycetes, but also in other important antibiotic producers belonging to the actinobacteria family. And when you have a closer look where they occur within the genome, then you can find them as part of biosynthetic gene clusters. And actually, as part of gene clusters that encode for different types of secondary metabolites like beta-lactam antibiotics, phosphonate antibiotics, polyketides, and so on. So therefore, we knew, okay, the subgenes are abundant in very different actinomycetes. They are part of gene clusters encoding for different types of secondary metabolites, and therefore we said that those ones are good candidates to be used as general activators of antibiotic biosynthesis. And then we tested this out in a random strain in just a running project of ours, and this also will be shown, I hope, in a few seconds, because back then this was a project which we performed with some Indonesian partners. So actually several ones that are sitting in the audience today. So with Puspita Listianti, Vin Kusarioto, Iran Dayan, Shantaratna Komala, and so on. So back then we aimed to isolate new antibiotics from Indonesian actinomycetes. So yeah, there we are. Back then, the Indonesian colleagues isolated up to 422 actinomycetes from Indonesian habitats. A subset of strains was handed over to our labs back then at the University of Tübingen, and we prioritized the strains for the most phylogenetically unique ones. We grew them in several different cultivation media and covered all of that to bioactivity tests to prioritize the one with the best bioactivity. And back then, 9 strains were of most interest, and from these ones we isolated the genomic DNA of, did biosequencing, and then performed a combined genome mining molecular networking approach. And I think this was what Ira mentioned some hours ago in her talk. So this we have done for dereplication purposes and focus on the most unique gene clusters that encode for the potential unique substances. And back then, one of the interesting strains that turned out from this analysis was SHP-227, which showed quite some good activity. And we just randomly used that strain and performed our sub-guided activation strategy with that. So we cloned in our PAPR2 overexpression strain to SHP-227 and got extracts of the cultivation of the strains and tested it against Bacillus subtilis as a test organism. And as you can see in here, the SHP-227 PAPR2 overexpression strain showed significantly improved bioactivity against Bacillus subtilis in comparison to the control strains with the anti-vector or the wild type samples, which means that PAPR2 overexpression led to significantly improved bioactivity of that SHP-227 strain. Now, what you would do in the next step is that you go into the lab and do elaborative isolation of that compound and check what the substance really is. But we didn't do that. In the first place, we started a genome mining approach of that strain in order to find out what might be the substance that encodes for this, or what might be the cluster that encodes for the substance with this improved bioactivity. And as I said, we had the genome sequence in hand. We performed an anti-smash analysis for the prediction of potential antibiotic gene clusters, and thereby we identified 25 potential secondary metabolite gene clusters. And then the question was, which is the one that encodes for this bioactive substance? And then we ran an additional bioinformatic analysis by applying the program PET scan and searched for our PAPA2 consensus sequence. And thereby we found this subtype sequence in front of gene cluster 9. And gene cluster 9 encodes for potential nucleoside antibiotic, which has a similarity to a gene cluster from a Streptomyces lineaceus strain. And that one is known to produce the substances amicetin and plicacetin, which are known compounds. They act as protein synthesis inhibitors with quite good bioactivities. And only when we knew that, then we went into the lab and checked by HPLC analysis what the product of this strain really is. This should be shown in the next slide, hopefully in a few seconds.
Sorry, Yvonne, I'm interrupted. Your time is 3 minutes again.
Okay, I will hurry up.
Sorry.
And I will go through the slides. Okay. So back then, we indeed could show that it's amicetin plicacetin that is produced by the PAPA2 overexpression strain and in significantly higher amounts. In the next slide, it is shown that this also helped for the isolation of 4 novel pyrimidine nucleoside antibiotics, which was a collaborative work with the group of Harald Groß at the Pharmaceutical Institute of the University of Tübingen. I'm really sorry, but the slides are just too slow. So this was just published in the beginning of this year. And now we know that this sub-activation strategy really works out. We are now having a set of different overexpression plasmids. I will skip that, but I hope, yeah, I hope that will work. So what we are just doing at the moment is that we apply this to the genome sequenced strains of the DSM-C. So we scale that activation strategy up, and we already screened hundreds of genome sequenced DSM-C strains for the abundance of sub-genes and sub-consensus sequences. And these are the ones that we are currently using for performing our sub-guided activation strategy. And in one of the next slides, hopefully, I still have the time that also, I will skip that one, that also in our selected DSM-C strains, where we clone in our PAPA2 regulator, we observe significantly increased bioactivities in comparison to the control strains. So when we test different test strains as for the bioactivity. And for some candidate strains, we already have some potential substances that are produced. For several strains, you can already see by the eye that something is going on, that when you clone in the PAPA2 construct, that there's significantly production, or that there's production of a pigment in comparison to the wild type strain. And we are currently isolating these substances and trying to find out if there's a novel antibiotic that is produced by those strains. And with this, I'm at the end. I'm really sorry that the slides were kind of delayed, but I would like to thank all the people that have been involved in this project, mainly Ira Handayan, who worked a lot with these Indonesian strains, especially with the SHP27 strain. Also, Janina Krause performed a lot of these sub-overexpression analysis, and currently the right-hand person are involved in the sub-guided activation strategy of the DSM-C strains. And with that, I would like to thank for your attention. I hope we can move on with joint collaborations because I really enjoyed that interaction with you. Okay, so thanks, and I would be happy to take questions later on.
Okay, thank you, Yvonne. Thank you very much for giving such an informative and exciting presentation. Now let me invite our second speaker, Professor Claudio Tiribili, MD, PhD.
Here I am.
Okay. Professor Tiribili, okay. Okay. Wait a minute. Okay. 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 Thank you. Professor Tiribili is the scientific director at the Fondazione Italiano Fegato. He has
Italian Liver Foundation. That's easier.
Oh, okay. Sorry. He has experience as an editor in the Journal of Hepatology and the Annals of Hepatology Journal, and experience as an associate editor in the BBC Research Note Journal. He is interested in research in bilirubin, neonatal jaundice, metabolic liver diseases, hepatocellular carcinoma, stem cell in hepatocellular carcinoma, Klicker Nazar syndrome, and translation medicine. Now allow me to welcome our second speaker, Professor Claudia Tiribili, MD, PhD, to make his presentation. Professor Tiribili, time is yours to give a lecture now. Thank you.
Well, can you hear me?
Yes, of course. We can hear you.
Thank you very much for this kind introduction. Let me try to share the screen with you. That's already done. And so I can start my presentation. Let me tell you, it's really a pleasure to be a speaker of the 9th International Symposium on Innovative Bioproduction in Indonesia on biotechnology and bioengineering. What I'm trying to address today, I'm driving the presentation. What I'm trying to do today is actually make a link to what we are eating and drinking and the liver damage. As you know, no, it doesn't work. So why don't we remove my presentation? I'll share my screen.
Okay. Stanley, can you help me?
Yeah, I mean, I need to go on. Let me try to share my screen. And then, here we go. Okay.
So it's moved now?
Yeah.
Okay.
Can you see it?
Yeah,
okay.
I'm seeing it now.
Okay, let's move back to the start. Sorry for that, but okay. So liver is, after the skin, is the largest organ in our body, and in an adult weighs about 1.5 kilo. Although it's not that thick, the normal liver has a volume similar to the soccer ball. And during this period, we are very fan of a soccer ball because it's the largest gland in our body, and 80% is composed by the liver, by the hepatocytes. But most important, the liver is the organ in our body which is the largest number of functions. And liver can do more than 600 functions, such homeostatic, metabolic, detoxificant, and immunological function. But in addition to that, and not only a marginal issue, is that the liver is the only organ in our body able to reproduce and regenerate. And this is a Prometeo myth is being depicted. The main causes of chronic liver disease are different things, such as diet, here, medication, viruses, and autoimmune liver disease, moving from normal liver to usually fatty liver, which is increased presence of the fat in the liver, which can go on with the steatohepatitis and then go to liver cirrhosis, which is advanced liver fibrosis and functional complication, ending up with the strip with the presence of hepatocellular carcinoma, which is the end stage liver disease. We need to stop this trip from here to here. And what I'm going to address today mainly is the role of eating and the diet in having liver disease. So And this is why, because MAFL, the so-called metabolic fatty liver disease, this associated metabolic liver disease, has a prevalence globally of more 25% of the population, which means in one out of the 4 European citizens has a fatty liver, so has a possibility to develop a serious liver disease. So if we go from the trip of from normal liver to hepatocellular carcinoma goes via different steps. First of all, we have the MAFL, the non-alcoholic liver, alcoholic fatty liver disease, which is, mainly represented by the increased amount of fat into the liver cell. But once the inflammation occurs, due most probably to oxidative stress, we have a sort of activation of the extracellular matrix of the liver, which is able to create scares and then move from NASH to cirrhosis, which is, labeled by the presence of scars in the liver, making the liver hard and less active. And these scars, together with the regeneration of the liver, end up in the hepatocellular carcinoma, which, as I told you, is the end stage of this trip. We have several risk factors for this. Of course, we have the overweight, the obesity. I'm going to touch this in more details later on. We have type 2 diabetes and insulin resistance. Altogether, overweight or obesity, type 2 diabetes, and insulin resistance make what we know now is called the metabolic syndrome. And the metabolic syndrome is related both in adults, but even more worrisome, is present also in children. And this is something we have to think about, because being in children, we need to prevent the fact that our child moves from year to year in the life span he or she is expected. And if we look here, that's very remarkable figure, that if you look the prevalence of obesity from 1975 to 2016, both in females and males, you may see the dramatic increase in the prevalence of obesity during this year, meaning that the more so-called civilized we are, the more fat we are going to be. And this is something we have to cope with and we have to fight with. And actually, even more worrisome is the trends in the number of children and adolescents with obesity. And if you both in males and in females, this is true all over the world. But if you calculate here, which is the Southeast Asia, also in the Southeast Asia, particularly in the male children are associated with an increased level of fat in the body. And if you look around the world, the presence of the metabolic fatty liver is basically almost the same all over the world. In some regions it's higher, like in US, in South America. In some regions it's lower, it's Africa, due to the fact there is a reduction in the calories available. Australia is basically like Europe, but in China we are observing now a booming wave of obesity, and this is something we have to cope with. So going back to what, in my opinion, is the most important aspect of the obesity is the so-called vicious circle of childhood obesity. We have a healthy child which actually due to a wrong life habits, sedentary life, lack of physical activity, is getting mildly obese child. And the mildly obese is actually a child is less prone to exercise, and those goes from mild to moderately obese child. And if this is also keep going with a wrong diet, is going to move from moderately obese child to severely obese child, and actually to and then to obese adult. And this is where we are going to go from a healthy child to a potentially sick child with the presence of fatty liver. And if you look around, you see that genetics may play a role in obesity. And we know now that the contribution of heredity to the risk of obesity is 25%. And this variability is related to the genetic background of the different studies performed. So we need to understand that habits, in addition to genetics, is the main predisposing factors, particularly habits of parents living with a child. And we just published an editorial in an international peer-reviewed journal, it was APT, making clear that it's the parents giving us the wrong information how to behave, less exercise and much more sedentary life. What are the targets of fat accumulation in the body? Of course, obesity is a multi-organ and multifactorial disorder. We can have that damage in the liver, in the brain, in the heart, in the vessels, in the nervous system, and in the pancreas, and in the muscles. All together, meaning that we have a lot of complications of obesity in the childhood. We have damages in the behavior, psychosocial, neurological, like pseudotumor. We have pulmonary, sleep apnea, asthma, and intolerance of the exercise. Of course, cardiovascular, due to the dyslipidemia, hypertension, coagulopathy, gastrointestinal, as I told you about steatohepatitis, and gallstones, renal, endocrine, and muscle. So this, we have to conclude that an obese children, like an obese adult, are sick, and we need to cope with. So which are the main causes? Is the imbalance between the calorie taken and the calorie expended. I'm used to say that the weight is like our bank account. If you eat, if you gain a lot of money, you don't spend, you get rich. But if you eat a lot of calories, but you spend a lot, you get poor. So we need to understand that globally, there's a striking decrease in physical activity. 1 in 5 boys and 1 in 10 girls is not having a real and appropriate exercise. And even more worrisome is that the amount of fruit eaten by our children is drastically reduced as compared to the ideal amount. Only 30% of boys or roughly 40% of girls eat a fruit a day, in spite of the fact that fruit is readily available all over the world. And the major suspect is sugar. Sugar, the white sugar, it's really being associated with the presence not only of obesity, but also liver disease. And the question is how much is too much? And we need to understand this because we can give advertising. And here is what we have. And what we have here in the slide is coming from the World Health Organization, telling us the content of teaspoons for each beverage. And mind that the WHO recommends consuming no more than 12 teaspoons per day. If you drink a Coca-Cola, you drink it's like you eat 8.5 teaspoons of sugars. If you eat a yogurt drink, it's like 7. But if you need an energy drink, you have 10. And of course, telling you that what we are using to give our children, the so-called sodas, are then maybe very dangerous. And here's what we have in as comparison. Of course, the blank is the water. There's no sugar. But if you look at Sprite and Coke, which is widely used, we have that 33.7 and 35 grams of sugar per each can. And even worse are Fanta and Schweppes when we go from 33 to 43 and 44 gram of sugar. So telling us that, we are not surrounded by a friendly beverage consumption. What is the total fat? The total fat should not exceed 30% of the total energy intake. And we have different kinds of fat. We have trans fats, the dangerous one, due to the so-called junk food. We have the saturated fat, which is related to coconut, palm oil, and alimentary, polyunsaturated skin, meat, and fruit. And we have then the unsaturated fat, which are the real good fat, that should be much higher than 90% of the total fat in intake we have every day. And this is important to prevent additional damage. How the excess of fat?
Sorry, Professor Tribali, I may interrupt you because your time is only 4 minutes again.
My time is still 5 minutes.
Oh, 5 minutes. Okay, 5 minutes again.
I mean, according to my watch, it's still 5 minutes.
Okay.
So I'll be on time. Don't worry.
Yeah.
So how the excess of fat and sugar impact the liver? This is a cartoon. It's a very complicated cartoon. I've had no time, unfortunately, to go through. But the key issue is that insulin resistance and increased intake for the diet bring together the possibility to accumulate fat in our liver. And this is what we have to deal with. So do we have animal models to study the juvenile fatty liver disease? The answer is yes. This is work we have done in our lab, driven by Sylvia and Natalia and Pablo. And what they did, they reproduced the fatty liver, a juvenile fatty liver, in a mouse, giving the mouse a lot of fat and a lot of calories. And what we have, what they have been able to reproduce is a fully scaled presence of fibrosis, meaning telling that this may be a good model to deal with in order to understand if there is any good treatment to work with this. In addition to that, we know that fatty liver is really linked to a leaky gut, because the gut is living alone with a lot of toxins moving from the gut to the liver. And this is what we have observed, that in large intestine, we have a leaky, the presence of a less tight junction as compared to controls. So in conclusion, we can say that excessive sugar and fat since young age induce obesity-related multifactorial events that lead to progressive multi-organ dysfunction, fibrosis, leaky gut, altered glucose homeostasis, which means diabetes. And this is, and we have to realize that the progression in children is much faster than adults. So we need to take care of this aspect for the future generations. And we know now that in the future 20 years, all the liver transplantation will be related to fatty children at the moment. So we need to take action on that. So let me skip all this because I have no time here. That let's try to deal with the future perspective. We heard that insulin resistant, type 2 diabetes, and obesity is the accounts for metabolic syndrome, which is a booming of obesity. And today we are still missing something, but we need to do something as a diagnosis, screening, and efficient treatment. And the clock is still ticking. So thank you for attention. I think I did my job in time, and I used all my 20 minutes.
Okay, thank you very much, Professor Tribali. Thank you for giving us such an informative and interesting presentation. And ladies and gentlemen, now are the time for the question and answer session. We have 20 minutes for Q&A session. If you have question for both speaker, please raise your hand and or you can write the question in the chat room. Okay, I think we have wait a minute. I need to see. I think Miss Atika, are you ready to give your question?
Thank you, Dr. Yeah, my question is to Dr. Yvonne. She's gone. No. Yeah.
Okay.
So,
Yvonne, are you still in here?
I'm still here. I'm here.
Okay.
Yeah. All right. Okay. So my question is a bit practical, no, but I never done the analysis that like what you did. So my question is like how long actually to get from your analysis of biosynthetic gene cluster until the analysis of the mutant, if I'm not mistaken. It's like you did like cloning for all this experiment, right? If I'm not mistaken. So ideally, how long until you get the results? Thank you.
Yeah, thanks for the question. That's actually hard to say because it's mainly dependent on the strains to manipulate. Actinomycetes are not so easy to genetically manipulate, and depending on which kind of organism you have in hands, this really depends how fast you are. But concerning the whole process, depending if you have the genome sequence, this is the first step. Let's say if you have the genome sequence, then you could go for the anti-SNP analysis, and this is quite fast because you just upload the sequence on the anti-SNP tool, and then you directly get the output after a few minutes. But this is, let's say, pure bioinformatics. It can give you an estimation what kind of gene clusters may be encoded, but you then need to really have a closer look into that and study that more in detail and also manually. And then, I mean, nowadays we have these overexpression plasmids in hands, they are cloned, then you just need to introduce them into the actinomycetes. And if everything works well, you are maybe successful after 1 or 2 weeks. If it's a strain that is harder to treat, then it can take several months, I would say. But this is what where I skipped a slide, because what I wanted to mention is that nowadays we have different genetic tools. So we have different plasmids, which can be used either for protoplast transformation, which is a process how you can get DNA into the strains, or you can use the plasmids for conjugation, where you use the opportunity that you have an E. coli strain that can deliver the plasmid to the actinomycetes.
Okay, Yvonne, thank you for the answer. And in the chat room, I have 3 questions for Professor Tribeli. First question is, it is true that some herb like curcuma is help in maintain our liver health? Thank you. This is from Mr. Elok. Miss Elok, I think. Are you ready to answer?
Yeah, the answer is maybe. I mean, there's no real confirmation that any drug is preventing liver disease. There are some drugs used to or suggested to be helpful, but I mean, the real scientific demonstration is not yet. The curcuma is one. The other one is silymarine, the thistle syrup. Others are the extract from the apple. But I mean, there are some information, but not real specific and solid scientific demonstration. So we need to take this with a very, with caution.
Okay, thank you for the answer. Okay,
Second
and the second question is, what are the most recent and reliable treatment to deal with liver damage that are already on cirrhosis stage? Is surgery and transplantation are the only option?
Well, it depends about the stage of the disease. Of course, if you have an end-stage liver disease, transplantation may be an option. Actually, it is an option. We have to realize that in Europe, every year we are performing roughly 2,500 liver transplantation all over Europe. But I mean, the ideal is not to go to the liver transplantation. The ideal issue is to stop the trip before moving ahead. This is what I try to demonstrate in my presentation as far as obesity is concerned. If we start changing our lifestyles by asking our adolescents and ourselves to exercise more, to eat less, and to be much more pragmatic in terms of daily behavior, this could be very helpful. Not only for the liver, but also for the cardiovascular disease, for diabetes, for everything.
Okay, thank you for the answer. And next, are there any possibility to recover from any damage in the liver?
For what? For?
Are there any possibility to recover from any damage in the liver?
Absolutely. I mean, liver is a regenerating organ. If you have a acute hepatitis, I mean, and you heal from the hepatitis, I mean, like a hepatitis, the liver returns back totally to normal structure. So we don't have to have any scar. Of course, if you don't recover fully, for instance, from B hepatitis or C hepatitis or other diseases, then the situation is not acute, it's going to be chronic. So the damage is chronic. So the end result is that the liver is daily damaged by the heating stimulus. And this ends up with something which is chronic liver disease.
Okay, thank you for the answer. And now we still have 13 minutes left. If there are any question for both of speaker?
We are being too clear.
Okay.
Or too confused.
Okay.
I have no idea.
If there any, if there's no any question again, I will ask maybe to Professor. Okay, Professor Mas. I want to ask Professor Iwan, what medium was used for self-activation with genome sequence in DSMZ strand? Is there any effect of the medium on the successful use of the self strategy in activating the silent gene cluster?
Yeah, actually, also here we tested a different set of cultivation media just to make sure that we do not miss anything. So actually we tested 3 to 5 different cultivation media. Some of them you may know, I will not go in detail with these ones, but it turns out that if you have activation, that 1 or 2 cultivation media are enough. So it looks like that really the abundance of the subtype activator is more relevant than the media you use.
Oh, I see. Okay,
okay,
thank you, Yvonne. Yeah,
but Ira, I think I also see a question in the discussion panel or in the chat room
I will check
from Hatati. I may also read it by myself because the question was what kind of data that one inputs to get biosynthetic gene cluster using Plantis mesh. So I'm not sure if this is if I understand the question correctly, because there are different tools available. So AntiSmash is the program that can predict antibiotic biosynthetic gene clusters encoded in bacteria, and PlantisMesh is a tool where you can identify gene clusters from plants. So if this question really targets PlantisMesh, and the question is which kind of biosynthetic gene cluster one can find there, I would say that you get an output which mainly contains gene clusters encoding for terpene substances, type 3 polyketide gene clusters, alkaloids, and so on, because these are more substances or gene clusters that are more abundant in plants. In bacteria, you may get just another output also on gene clusters encoding for non-ribosomal peptides, type 1, type 2 polyketides, for example. So I hope I could answer that question.
Okay. Yeah, thank you, Yvonne. If there are any question again for the both speaker, please raise your hand or write it in chat box. Maybe I will ask Professor Claudia Terribili. Does consuming too much fiber also causes liver damage?
Yes. I mean, the point is not what you consume. The point is what the amount you consume and the calories. I mean, if you eat, let's say, a kilo of bread, the amount of calories is much higher than a kilo of salad. But of course, the taste of 1 kilo of bread is much better than the taste of 1 kilo of salad. And if you put some butter on your bread, the calories increase. So I think we need to make a balance of our daily behavior. I mean, if we eat more calories, we need to spend more calories. As I told you during my presentation, our weight and our metabolic functions is like the bank account. I mean, if you gain a little amount of money, but you are so able not to spend, you're getting rich. But if you gain a lot of money, but you spend a lot, you spend more, you get poor. So everything is balanced. We need to be careful. And then the basic point is to teach our children to behave properly. And our children, at least in Europe, don't behave properly. They are in front of a screen. They don't move. They don't go to walk. They don't go to play football. They don't go on the bike. They stay home, and this is bad. That's why we're having a real problem with it with adolescents.
Okay. My next question is there any effect of a supplement on the liver health?
You mean the supplements?
Yeah, like vitamin?
Well, I mean, the supplements have never been demonstrated to be helpful. The supplements are very helpful for the companies that are making the supplements. They're making a lot of money they're making a lot of good for the producers of the supplements, but there is no evidence that they're making good for the liver of the subjects taking the supplement.
Okay, thank you.
So take a message, don't take supplements.
Oh, okay, thank you. Thank you for your suggestion.
Eat the fruit, it's more than enough.
Oh, I see.
You're lucky enough to live in a country like Indonesia, where the fruits is so beautiful. And you have mangoes, you have everything.
Yeah.
Why don't you have to take supplements? If you're living in Europe, I mean, you go and then eat an apple or an orange or a pear, and that's it. It's better, it's cheaper, it's much more healthy. Trust me.
Yes, of course. Thank you for your suggestion. Okay, if our time is left 2 minutes. Okay, or Yvonne, do you want to ask the question to Professor Tribble?
Yeah, actually, I would also want to ask a short question to Mr. Tiribelli. I wondered if there are any numbers. What is in general more harmful if one drinks too much alcohol, or using too much calories from early days on?
I didn't touch alcohol for obvious reasons, because but alcohol, there is a safe amount of alcohol daily, which around 30 grams per day, both in females and males. 30 grams of alcohol per day means half a liter of wine or a small glass of super alcoholic schnapps or whatever it is, which means that if you have one glass of wine with your meal, it's good. What is bad is to drink outside the meal, because the absorption of the alcohol without any aliment is very quick. This is why the heavy drinkers are used to eat together with alcohol something which is oily, like olives and peanuts, because you have a sort of film of oil in your stomach, so the absorption is less, and then you can drink more. That's the trick they're doing. So, a limited amount of alcohol is safe, actually. But, it depends on how limited is limited. And, a good glass of wine is good. And let me tell you that, unfortunately, I'm a teetotal. I cannot drink alcohol, not because I don't want, because I cannot stand it. And this is my limitation, because, with a good meal, a glass of wine, assuming that your religion is allowing to do it, it's good.
Okay, thank you for the answer. There's 3 questions again in the chat room. First, for Professor Tribble. The question is, as we are getting older, is it true that our metabolic rate are also slower than when we are young?
Yes. Yes, the answer is yes.
Yes, okay.
That's it.
Yeah. And there's 2 questions for Yvonne. Did you also conduct any research in Germany on cool culture to activate a gene cluster for the production of natural products? based on your experience, which substances from terrestrial and marine ecosystem produce more new metabolic at this time?
Okay, concerning question 1, we didn't do co-cultivation approaches, and I tell you why. Because later on, the identification of the produced metabolites is just too hard. If you do co-cultivation, you may activate something, but you do not know what is produced. And then you need to go again into the lab, isolate the substance, do chemical analysis to really identify the produced substance. We had the attempt to do a targeted activation, because if we use these kind of regulators, then we exactly know where they bind, which kind of antibiotic gene cluster is activated, and this facilitates later on the identification of the compound. And therefore, we didn't do co-cultivation, which also may work, but we only focus on this targeted activation approach. So the second question is, which kind of organisms do produce more antibiotics, terrestrial actinomycetes or the marine derived ones? We cannot say until now. You can have marine organisms with really a lot of antibiotic gene clusters, but also terrestrial ones can harbor more than 50. So I don't see a difference in terms of the potency of terrestrial against marine organisms. So there's still enough out there to be identified in terms of new secondary metabolites, I would say. Okay, but I think there's an additional question. Ira, would you like to?
Okay, I think there is no any question again. Okay, I think time is finished. Time is the end for the question. Finally, we come to the end of the seminar. I want to draw a conclusion from what speaker said. From Professor Yvonne Mas, we can learn that a self-guide strategy can be used to activate antibiotic biosynthetic gene cluster in a targeted manner and thereby exploit the biosynthetic potential of actinomycetes. And from Professor Tribuli lecture, we can conclude that excessive sugar and fat consumption from a young age can lead to the obesity, leading to the development of metabolic associated fatty liver diseases and non-alcoholic fatty liver diseases in the children. Unfortunately, the development of metabolic associated fatty liver diseases in the children is faster than in adults, and efficient therapy to treat this disease doesn't yet exist. Hence, the best options still depend on the lifestyle changes. Okay, finally, I would like to thank to Professor Yvonne Mas and also Professor Claudia Tribuli for their informative and interesting presentation, and all the participants for their very active participation. And now I will hand over the event to the MC. Thank you. Ms. Aulia, time is yours now. Thank you.
Terima kasih.
Terima kasih banyak.
Thank you, Professor Tribuli, Professor Yvonne Mas, and also Dr. Ira for the amazing and also presentations this afternoon. Distinguished guests, ladies and gentlemen, now we have arrived to the end of today's conference. On behalf of official committee, I would like to thank you for your attendance and also active participations on the first day of ISIBio 2022. We're signing off. See you on the second day of 9th ISIBio tomorrow at 9 AM Jakarta time with another interesting speakers and also materials. Stay safe and also healthy.
Bye-bye. Thank you.
Thank you very much, Yvonne and Professor Tribuli.
Thank you. Bye.
Bye.
Bye-bye.
Bye-bye.